Have you ever wondered how a single bacterium can, well, basically build an empire? It's not magic. It's a story of multiplication, and it's happening on a scale that's just mind-blowing. So let's dive into the super elegant and crazy efficient process that makes it all possible, binary fission.
Right, so how does one tiny cell build an empire? When we think about growth, we usually think of something getting bigger, right? But for microbes, that's not the whole story. It's all about an explosive increase in numbers. We're talking going from 1 to 2 to 4 and then, well, to billions. It's multiplication on a scale that's almost hard to wrap your head around.
Okay, so at the very heart of this incredible story is a simple idea. One cell becomes 2. For bacteria and their cousins, the Archaea, this is pretty much their go-to method for making more of themselves. It's super straightforward, super efficient, and it's called asexual reproduction.
And that brings us to the star of our show, binary fission. The name really gives the game away, doesn't it? Binary, that's 2. Fission, that means to split. That's literally all it is. The cell just grows a bit, copies everything inside, and then splits right down the middle. And boom, you've got 2 perfect identical clones. Now, to really get why this is so cool, let's think about how our cells divide. Mitosis? It's this whole complicated, carefully choreographed dance with a nucleus and all these pairs of chromosomes. Binary fission, on the other hand, it's just beautifully simple. It's way faster, way more streamlined, mainly because it doesn't have to deal with all that complex machinery.
But what actually kicks this whole thing off? I mean, a cell can't just decide to divide whenever it feels like it, right? There's got to be some kind of trigger, some signal that says, all right, it's go tad.
And here's what's so clever. The trigger isn't something from the outside world. It's the cell's own growth. As the bacterium gets bigger, it starts building up this key protein called DNA. And once it hits a certain amount, that's it. It's the green light. The whole division process gets kicked into high gear. It's such an elegant system, isn't it? It directly ties the cell's size to its decision to make more of itself.
And this whole process, well, that's what we call the bacterial cell cycle. It's just this continuous, beautiful loop. A new cell is born, it grows, that growth triggers it to copy its DNA, it divides, and then the whole cycle starts fresh with its 2 new daughter cells, over and over again.
So the green light is on. The cell is committed. Now it has one absolutely critical job to do first. Before it can even think about splitting in 2, it has to make a perfect, flawless copy of its genetic instructions, its blueprint for life.
And a huge reason this is so efficient is the simplicity of the bacterial chromosome itself. See, unlike us with our multiple, long, stringy chromosomes, a bacterium usually just has one, and it's a single closed circle of DNA. The best way to think of it is like a single-chapter instruction manual that's been looped into a circle. It has a very clear starting point for copying, called the ORIC, and a clear endpoint, the TER.
Okay, so to actually make that copy, the cell builds this incredible piece of molecular machinery right at that starting point. This whole protein crew is called the replication. You can think of it as the cell's high-tech photocopier. Its only job is to latch on and zip along that DNA circle, reading the original blueprint and building a brand new, identical one as it goes.
All right, let's just take a step back and look at the big picture here. You can really break it down into 3 main stages. First up, you've got the duplication of the chromosome. That's the copying we just talked about. Then, while that's happening, the cell itself starts to stretch out and get longer. And finally, the last step is the actual division, where it splits into those 2 separate complete daughter cells.
Okay, so now we have a problem. Well, a logistical problem anyway. The cell has 2 perfect copies of its instruction manual. But how does it make sure that one copy goes to one new cell and the other copy goes to the other? It can't just be random. It needs a system to actively pull them apart.
And this is where our movers come in. Meet proteins ParA and ParB. These 2 work together to form this amazing little molecular pulley system. Their whole job is to grab onto those copied chromosomes and physically drag them to opposite ends of the cell. This makes absolutely sure each new daughter cell gets its own complete instruction manual.
So here's how they do it. Step 1. Near that starting point on each chromosome, there's this special little docking site called ParS. The first protein, ParB, zooms in and latches right onto that site on both of the new DNA copies. It's like grabbing the handle.
Now, once ParB has a firm grip on the DNA, it then grabs onto this long filament made of our other protein, ParA. And this interaction is just like reeling in a fishing line.
It actively pulls that entire chromosome copy all the way over to the opposite end of the cell. It is such a beautiful and precise little mechanism.
All right, let's recap. The chromosomes are separated, one at each end. The cell is stretched out. We've officially reached the climax of our story, the grand finale, the final split.
This final act has a name. It's called septation. And really, all it means is the process of building a new wall, or a septum, right down the middle of the cell. This is the wall that's ultimately going to separate it into two new, distinct individuals. And this isn't just a simple pinch off. It's a really carefully managed four-step construction project. First, the cell has to find the exact middle. Then it builds this amazing contractile belt of proteins called the Z-ring. Next, it calls in all the construction workers, the machinery needed to build a new cell wall. And finally, that Z-ring starts to tighten, like a belt, pulling that new wall inwards and pinching the cell cleanly in two. And there you have it, the end result of this whole elegant process. Two new daughter cells. They are perfect genetic clones of each other and of the parent cell that started this whole journey. And what are they going to do now? Well, they're ready to start growing, building up their own DNA, and starting this amazing story all over again.
But here's the part that just blows my mind. The speed. For a common bacterium like E. coli, if the conditions are just right, this entire story we just told, from one cell becoming two, takes just 20 minutes. 20!
So, I'm going to leave you with a little thought experiment. If one cell can become two every 20 minutes, what does that look like after a single day? Think about that for a second. The numbers get astronomical, fast. And that right there shows you the incredible, mind-boggling power of binary fission. Thanks for diving into this with me.